Inside your body, the immune system is constantly trying to answer one critical question: does this belong here? Every day, it encounters bacteria, viruses, toxins, damaged cells, and harmless molecules from food or the environment. Antibodies are one of the most precise tools it uses to sort those signals. An antibody, also called an immunoglobulin, is a large protein made to recognize specific targets called antigens. These targets can be parts of microbes, fragments of toxins, or structures on infected cells. The antibody's job is not simply to float around and attack everything. Its real power is recognition. It can identify a meaningful molecular detail, bind to it, and turn that hidden threat into something the immune system can clearly see and respond to. Antibodies are often shown as Y-shaped molecules, and that simple image tells you a lot about how they work. At the two tips of the Y are regions that bind to antigens. These binding regions are called paratopes. The exact part of the antigen they attach to is called an epitope. The relationship is highly specific, almost like a molecular grip fitting onto a particular surface. This does not mean every antibody recognizes an entire virus or bacterium. Often it recognizes one small feature on that larger object. A virus may have many possible epitopes on its surface, and different antibodies can bind to different ones. This is why antibody recognition can be both extremely precise and incredibly varied. That precision matters because the body has to recognize an enormous range of possible threats. Antigens can come in many sizes and chemical forms. They may be proteins, sugars, or other molecular structures. The immune system cannot rely on one generic detector for all of them. Instead, antibodies exist in a huge variety, especially at their binding tips. Those variable regions allow different antibodies to match different epitopes. One antibody might recognize a structure on a bacterial surface, while another recognizes a viral spike, and another binds a toxin. The immune system's challenge is scale. There are millions of possible targets. Antibodies meet that challenge by creating a vast library of molecular shapes capable of recognizing very different invaders. Once an antibody binds to its target, it can protect the body in more than one way. One of the simplest and most direct methods is neutralization. Imagine a virus that must use a specific surface structure to attach to a human cell. If an antibody binds over that structure, the virus may no longer be able to enter. It has not necessarily been destroyed yet, but it has been disarmed. The same idea can apply to toxins. If an antibody covers the part that interacts with cells, the toxin may lose its harmful effect. Neutralization is a major reason antibodies are so important in protection after infection or vaccination. They can stop a threat before it gets a chance to spread. Antibodies can also work by tagging targets for destruction. A bacterium or infected cell may be hard for the immune system to handle efficiently on its own. But when antibodies coat its surface, they act like warning labels. Other immune cells can recognize the antibody-covered target and respond more aggressively. This tagging helps immune cells engulf microbes, destroy infected cells, or activate additional defense systems. The antibody is not always the weapon that delivers the final blow. Often, it is the marker that tells other parts of the immune system where to focus. In that sense, antibodies are both identifiers and coordinators, connecting recognition to action. Antibodies can exist in different locations. Some are attached to the surface of B cells, where they act as B cell receptors. In that position, the antibody is like a sensor mounted on a living immune cell. If it binds the right antigen, it can help activate the B cell and start a larger immune response. Other antibodies are secreted, meaning they are released into the spaces outside cells and into body fluids. These free antibodies can circulate, search for matching antigens, and bind them wherever they are found. When people use the word antibody in everyday conversation, they usually mean this secreted version. The term immunoglobulin can refer to both the cell-bound and secreted forms. The immune system does something remarkable to create antibody diversity. It does not store a separate complete gene for every possible antigen the body might ever meet. Instead, developing B cells rearrange genetic segments to produce unique antibody-binding regions. This process creates a massive range of possible paratopes. Most will never be useful, but some will happen to fit real threats. When an antigen appears, the B cells with matching receptors can be selected and expanded. This is a core feature of adaptive immunity. The system does not merely rely on fixed defenses inherited at birth. It can generate diversity, select useful responses, and amplify the ones that match the current danger. Although the tips of antibodies vary enormously, the rest of the molecule is more stable, and that stable region helps determine what the antibody does after binding. In humans, antibodies are grouped into 5 main classes, also called isotypes: IgA, IgD, IgA, IgG, and IgM. These classes are not just different names for the same thing. They differ in structure, location, timing, and immune function. The binding region answers the question, what antigen is this antibody recognizing? The class helps answer the next question, what kind of immune response should happen now? This division of labor makes antibodies flexible. They can recognize countless targets while still triggering organized, specialized responses. Each antibody class has its own role in the body's defense strategy. IgG is common in the blood and is strongly associated with longer-term protection. IgA is especially important at mucosal surfaces, such as the respiratory and digestive tracts, where many invaders first try to enter. IgM often appears early in an immune response and can bind targets effectively in larger molecular arrangements. IgA is best known for its involvement in allergic reactions, but it also plays a role in defense against certain parasites. IgD is less famous, but it appears on B cells and contributes to immune regulation. Together, these classes show that antibodies are not one tool. They are a family of specialized tools. Antibodies are central to what is called humoral immunity. The word humoral comes from the older idea of body fluids, and in modern immunology it refers to defenses carried through fluids by soluble molecules like antibodies. This is often contrasted with cell-mediated immunity, especially responses involving T cells that directly kill infected cells or coordinate immune activity. The distinction is useful, but the immune system is not divided into isolated departments. Antibodies can recruit cells. B cells can receive help from T cells. Complement proteins can join the process. Humoral immunity simply highlights one major strategy. Protection can be carried through the body by proteins that bind specific threats and guide the response. Most antibody responses are considered part of adaptive immunity because they are specific, shaped by genetic rearrangement, and connected to immune memory. After the body encounters an antigen, some B cells can become long-lived memory cells. If the same antigen appears again, the response can be faster and stronger. This is one of the principles behind vaccination. A vaccine introduces the immune system to a harmless version, fragment, or instruction related to a pathogen, giving it a chance to prepare. Later, if the real threat appears, antibodies may already exist or be produced rapidly. The body is not starting from zero. It is drawing on a previous lesson. There are also antibody functions that blur the neat line between adaptive and innate immunity. Natural IgM antibodies, for example, can be produced without a typical targeted immune response and can recognize multiple unrelated antigens. This is called polyreactivity. These antibodies are not as narrowly specific as the classic picture of one antibody matching one epitope, but they can still be useful. They can work early, before a highly tailored response is fully developed, and they can cooperate with the complement system to help clear antigens and immune complexes. This shows that antibodies are not locked into one simple category. Some act like precise adaptive weapons, while others behave more like early broad response tools. Antibodies also reveal why details matter in biomedical research. Different species can have antibody classes with similar names, but those classes do not always perform identical functions. A mouse antibody labeled with one class name may behave more like a different human class in practical immune activity. That matters when scientists use animal models to study vaccines, infections, or antibody-based treatments. The general principles may translate, but the exact immune effects can differ by species, tissue, timing, and antibody class. So antibodies are not just generic immune proteins. They are context-dependent molecules whose structure, location, and class determine what happens after recognition. The simplest way to understand antibodies is to see them as molecular bodyguards with two linked jobs. First, they recognize. Their variable tips bind specific epitopes on antigens with remarkable precision. Second, they direct. Their more constant regions help determine whether a target is neutralized, tagged for cleanup, connected to other immune systems, or remembered for the future. This combination of specificity and coordination is what makes antibodies so powerful. They can block a virus, mark a bacterium, support long-term immunity, and help the body learn from past encounters. In a world full of microscopic threats, antibodies turn molecular recognition into organized defense. If you found this helpful, please like this video and subscribe for more explainers.